The objective of this procedure is to assess the microtubule organization and dynamics in vivo. Neurons are polarized cells with distinct axonal, dendritic, and synaptic compartments maintained by their underlying cytoskeleton. Microtubules form majority of the neuronal cytoskeleton, and their dynamics and orientation determine key events during neuronal development and maturation. Microtubules are comprised of alpha and beta tubulin heterodimers and are inherently polarized with highly dynamic plus ends and stable minus ends. During polymerization at the plus ends, a multimolecular complex of end-binding proteins transiently associate with the protofilaments and promote the assembly of tubulin dimers.
This technique will help us to visualize neuronal microtubules in vivo. Using this technique, one can determine the orientation and other parameters of dynamic microtubules during development and regeneration of C. elegans neuron. We typically use this reporter to visualize microtubule dynamics in mechanosensory neuron. However, one can express this reporter in other cell types such as muscles and skin to visualize dynamic microtubules in these cells.
Fluorescently labeled end-binding proteins like EBP to GFP here appear as comets. Dynamics of these comets has been correlated with concomitant growth of the microtubules and a key indicator to gauge microtubule dynamics and orientation. In order to observe these comets in a specific cell type, transgene with DNA of EBP2 and GFP are expressed under a specific promoter. For expression in the PLM neurons, this transgene is expressed under the MEC4 promoter. These transgenic worms can be viewed under a stereo